Hi everyone, I'm Zixiachen from CSBE. The literature I'm honored to share with you today is a major methodological article published in PNAS in 2025. It is titled Synthesis of Large Single-Transcript Pathways from Oligonucleotide Pools, Design of Starburst, an Autobioluminescent Reporter, though a brilliant computational design of multi-enzyme orthogonal isolation. This study solves a problem that has long plagued plant synthetic biology, the low-cost assembling of mass gene pathways. Before diving into the core technology, we must understand the three major bottlenecks currently facing plant synthetic biology. First, custom synthesizing long genes pathways is prohibitively expensive, which makes large-scale trial and error near impossible. While oligonucleotide pools are incredibly cheap, they are a chaotic mix of thousands of tiny 200 base pair fragments, making it a nightmare to accurately extract and assemble the specific pieces we need. Second, plant transformation relies heavily on agrobacterium, but once our plasmid size exceeds 30 kilobases, the delivery efficiency simply drops off a cliff. And finally, we face a severe shortage of unique plant promoters, forcing ourselves to reuse the same few promoters not only makes our vectors massive, but it inevitably triggers gene silence within the plant. To address the issue of promoter scarcity and vector overload, the authors introduced a polycistronic mRNA strategy. First, the standard monocistronic coding regions are common in most higher eukaryotes. In this genomic structure, protein coding genes are interrupted by non-coding introns, and transcription is regulated by specific promoters and enhancers. Therefore, the transcription of every single gene is independently controlled. A polycistronic system, on the other hand, uses just one single promoter to transcript an ultra-long mRNA, which is then translated into multiple independent proteins. While this is very common in prokaryotes, ribosomes in eukaryotic plants generally lack of the ability to continuously translate multiple independent proteins from a single strand. Because of this limitation, we must borrow a mechanism from viruses, the 2A peptide. The chemical of the 2A peptide is highly unique. During translation, it triggers a process known as ribosomal skipping. Specifically, it blocks the formation of a peptide bond between glycine and proline. This causes the upstream proteins to disconnect and release, while the ribosome simply continues translation the downstream protein. However, if you use the 2A peptide entirely on its own, the cleavage efficiency is often quite poor. This because of the large folding proteins on either side creates steric hindrance or spatial crowding, which leads to uncleaved giant fusion proteins. To solve this, the authors added an ALP4 linker right in front of the 2A sequence. You can think of this linker as a flexible spring. It relieves that spatial tension. Next, let's look at the core of this paper. How do they actually construct this massive sequence containing numerous 2A peptides and target genes? To achieve this, the authors developed an open-source automated assembling pipeline called EGPOP. So how are the sequences in the oligopool actually designed? First, the software removes any internal restriction sites and optimizes the GC content. It then divides the long target pathway into short fragments of no more than 300 base pairs. Crucially, the software also filtered out any four base pair overhangs that are prone to self-folding or are simply too similar to one another. At both ends of these fragments, specific index primers are designed. This allows to amplify the specific target double-strand DNA out of the massive oligopool. Next, they introduce the first enzyme, which cuts off those outer primer. Following a critical magnetic bed purification step to remove that waste, they add the second enzyme and the ligase, exposing the pre-designed 4 base pair overhangs. From there, the ligase acts exactly like a zipper and seamlessly stitching the fragments together in the correct order. This two-enzyme strategy is absolutely vital. It prevents the cleaved primers, the garbage, and our target fragments from sharing the exact same sticky ends. Without this design, the ligase would blindly reattach the garbage back onto the gene, leading to a complete assembling failure. Next, the authors validated their system. Using their built-in library of hundreds of orthogonal indices, they amplified and assembling 53 completely different plant transcription factors from a single pool. The sequencing results were impressive. 93% of the clones were structurally correct, and 69% had absolutely zero PCR mutations. Taking it to the extreme in Figure 2C, the authors challenged the system by assembling 80 tiny fragments into a 3.1 kilobase GFP in a single one-spot reaction. The numbers 1, 2, and 3 in the figures represent 3 completely independent clones, and all of them emitted strong fluorescence, which perfectly demonstrates that the system's incredible assembly robustness and reliability. Now let's look at the core challenge, constructing the 9.5 kilobase pair starburst pathway. You might wonder, why can't they just assemble all 50 plus fragments in a single tube? This is because of the physical limitation called overhang exhaustion. While there are 256 possible 4 base pairs combinations, only about 30 are truly high fidelity and won't interfere with each other. If they tried to assemble 50 fragments at once, it would run out of the unique codes and be forced to use very similar ones, leading to mismatch ligation. Figure 3a shows how a two-step hierarchical assembly solves this. In level 1, they grouped 6 fragments per tube for assembly use BPS1. This physical separation allows us to safely reuse the same high-fidelity cookbook to build 1,000 base pair modules. Finally, these modules are combined in a single tube using a different high precision set of codes and the BSMB1 enzyme to safely completes the 9.5 kilobase pair pathway, perfectly avoiding any assembly errors. After assembling the pathway, the researchers tested different arrangement of the 5 genes which you can see in Figure 3b. The difference in brightness between this arrangement was massive. So why did this happen? It reveals the ribosomal drop-off effect inherent to the 2A system. Ribosomal skipping is not 100% perfect. At every single 2A junction, there is roughly a 10 to 20% chance that the ribosome will simply fall off the mRNA. This means that on a long 9.5 kilobase transcript, the gene placed at the very front will produce the most protein, while the gene at the very end will have the lowest expression level. To create light, these 5 specific enzymes must be present in a perfect concentration ratio. By shuffling the gene order, the authors cleverly exploited this nature expression gradient. They screened through different combinations until they found the absolute perfect arrangement, the one that delivered the brightest luminescence and the optimal ratio of metabolites. Ruby is a plant reporter gene that produces a vivid red pigment. The researchers performed standard codon optimization on Ruby for Arabidopsis. However, the results were completely unexpected. The pigment production of these optimized variants plummeted by 5 to 10 times compared to the original sequence. Through in-depth analysis, they discovered the culprit. The original Ruby was optimized for rice, and rice genes naturally possess an extremely high GC content, up to 58%. Once it was recoded for Arabidopsis, the GC content crashed to 45%. Even when they painstakingly removed all predicted cryptic intron splicing sites, the expression level remained dismal. The conclusion here is quite profound. For long, artificially synthesized polycistronic transcripts, high GC content is the absolute core determinant for maintaining the mRNA secondary structure stability. Based on this, they used the DNA chisel to completely rescarve the starburst sequence, pushing its GC content up to between 53 to 63%. As you can see in the results, this GC-boosted starburst achieved brilliant naked eye visible luminescence in stable transformed Arabidopsis plants, perfectly validating their hypothesis. In conclusion, the authors demonstrated that the polycistronic LP4 2A system successfully shatters the bottleneck caused by the scarcity of plant regulatory elements. Furthermore, the EGPOP pipeline, through its brilliant 2-enzyme orthogonal isolation and two-step assembly, completely overcomes the physical limits of overhand exhaustion. This enables the ultra-low cost assembly of near 10 kilobase pathways directly from oligopools. We also learned the high GC content rule. Pushing GC content above 50% is the hidden crucial variable that dictates the success or failure of expression ultra-long synthetic genes in plants. But perhaps the most admirable aspect of this work is its final design. The authors intentionally flagged the entire 9.5 kilobase product with BSA1 restriction site. With a single enzyme cut, it generates the standardized AATG and GCTT overhangs. This means Starburst is not only a standalone project, it has been transformed into a true plug-and-play component, perfectly compatible with the global Moklo and Golden Bridge international standards. That's all. Thanks for your listening.